So let's move on to hear from Dr. Stephan Urban, who's with the German Center for Infection Research, Heidelberg partner site in Heidelberg, Germany. Stephan has done extensive work on molecular mechanisms of hepatitis B and hepatitis D virus-host interactions. And Stephan and his group developed bulevirtide, the first approved therapy for the treatment of chronic hepatitis D. And we're very fortunate to have Stephan join us today for his remarks. And Stephan, over to you.
Okay, thank you very much. I try to share my Okay, let's start. Go back. Okay, can you see my slides and can you hear me?
Yes.
Yes, we can.
Okay,
yes,
thank you very much. So thanks a lot first for the invitation. Thanks to John, and I want to give a very special thank to Ryan Koshi, who participated in putting the program together. Ryan was really one of the very first people I talked to him on HPV in 1988 when I was a practical student in Munich, and I wish him a very good retirement. I would like to give you an update of entry inhibitors to treat HPV and HDV infection. And let's start. If we look at an entry inhibitor, we look at both viruses in common because they share the envelope protein that you already heard of, the large, small, and middle protein. And the Delta virus uses these envelope proteins. Therefore, Delta virus is very special, not only as a pathogen replicating by a completely different mechanism. That means that it cannot be treated efficiently with nukes. But it can be treated as HPV may be in the future with an entry inhibitor. And the whole thing that we have to ask ourselves for almost 2 decades is how these viruses can efficiently target liver cells, hepatocytes, go into the liver and do that with a very, very high efficiency. We all know that very low doses of virus is enough to infect a liver for HPV or super infected liver that's already infected with HDV. And we did spend a lot of work in primary human hepatocytes because we were lacking infection system to find out that there are different infectivity determinants, the attachment and fusion, for example. This is the antigenic loop. This means that antibodies that are directed against these loops are not directly interfering with the receptor, but as we know with heparan sulfate proteoglycan, and that the Prius domain in the large envelope protein plays an important role in engaging a receptor that we now know is NTCP, but that was also not known for a very long time because we were lacking the systems to screen for that receptor. If you look at both viruses from a general point of view, they enter via heparan sulfate proteoglycan and NTCP. And then, as you already heard, they deliver they form the cccDNA in the case of HPV or a circular single-stranded RNA in the case of Delta virus. And they can amplify maybe within the cells. That's internal amplification, and an entry inhibitor would not affect these steps. But an entry inhibitor would affect steps that are referred to external amplification. So either super infecting the same cells or then spreading into other uninfected cells. And the question is, of course, to what extent do these external routes contribute to the maintenance of Delta or HPV? Cannot go into detail, but there seems to be a potent role for this mechanism of external amplification, and consequently blocking this effect has therapeutical effects. Now, if you look at entry inhibitors, I just want to mention that, of course, monoclonal antibodies targeting Pre-S and preventing infection have already been investigated by Gerlich's lab in 1984. They are very potent inhibitors, antibodies that either directly interfere with Pre-S binding to the receptor or with attachment, for example. But they are also like the HPICs, but there are also attachment inhibitors like heparin, suramine, salps, and they interfere with the engagement of heparan sulfate proteoglycan. But the NTCP, sorry, that's a typo, NTCP specific inhibitors like hepcludex or bulevirtide, formerly mercludex, but there are other cyclosporins have been shown, or substrates for NTCP that are transported by that receptor. They could act as entry inhibitors. And for example, bile salts, which are the natural substrates of NTCP, but also other transporters, they have been shown to inhibit infection. But we have very strong differences in the IC50, in the inhibitory activity. And I come to that in a moment why that is. And that's important for the development of entry inhibitor. Let's share with you some thoughts on the hepcludex and some structural details. The hepcludex or bulevirtide has been developed by an infection interference assay. So we wanted to know whether peptidic agents that are derived from the pre-S can interfere with infection. And the bulevirtide is a drug that is not representing completely a natural sequence, but it has been optimized. And you can see if you have these myristylated peptides that just represent this N-terminal part, you can profoundly interfere with infection. This is HPV infection, that's delta infection in hepar cells, but that also works in primary human hepatocytes. And the concentration that you need to interfere with infection are extremely low. That's really amazing. 80 picomolar in primary human hepatocyte. You're in the subnanomolar IC90 levels. And the drug also exclusively target the liver. I show you that in a moment, just a nice picture. And they are specific for NTCP. There's only residual binding to other bile acid transporters in the liver. So that's the consequence of the co-evolution of that HPV virus to its receptor. That's NTCP. It excludes other OATPs, for example, and that's also very important for using these inhibitors as drugs. If you fluorescently label such a molecule like the bulevirtide and you put it onto primary hepatocytes, they need to be differentiated. If they are not differentiated, there's no binding. Receptor is not expressed in HepG2 cells on its own. And the remarkable observation was that if you do a time course, these molecules remain on the surface of hepatocytes, as you can see here, for more than 18 hours. It means the half-life time is very, very long. And that's quite impressive for a receptor because normally it's within seconds or minutes that a receptor is uptaken into the cell. That also may explain the low kinetics of infection of hepatitis B virus. Another interesting finding, if you label radioactively these peptides and inject it into a rat, for example, they target the liver with an enormous efficacy, more than 95% within several minutes. And this peptide is bound to the surface of the hepatocytes on the NTCP. That picture was taken at a time when NTCP was not recognized yet. And if you mutate the peptide with just 2 or 3 amino acids, you get a completely different distribution of that molecule in the organism. We've also done that, of course, during the clinical development in chimpanzees, and you see a similar thing. So at that time, we were asking which molecule is addressed, and we all know now, due to the groundbreaking work also in Willy's laboratory, that it is NTCP, the bile acid transporter, and we have since recently the structure, the cryo-EM structure of that receptor on its own. And what you can see here, it is first an integral transmembrane protein sitting in the membrane, the basolateral membrane of the hepatocyte. It transports bile acid through that tunnel that it can form. And the interesting observation here is that there are obviously 2 bile salt molecules that fit into that may lead to the selection of the bile salts during the transport. Those that are not conjugated, for example, are spit out again. The others are transported. And there is a conformational change driven by a sodium gradient. Here you see the sodium ions that drive that molecule into 2 conformations and allow thereby the bile acid transporting. That's the natural function of that molecule. Now the big question is, how does that bulevirtide, or let's say pre-S NTCP complex looks like? Because that's not only the drug complex to the receptor, it's also, of course, that part of the pre-S domain to the receptor. And in very nice collaboration together with a Swiss group, Kaspar Locher, from the ETH in Zurich, this complex was solved. But you see here is the NTCP in green, and you see the bulevirtide, which on its own is not structured. So it just is a random peptide that does not contain or does not build a structure on its own. And you can separate them into a myristoyl anchor here in pink, and in a black domain, that's the first 20 amino acids, and a string domain, which is then from 20 to or 21 to 48. And if you look here, the myristoyl anchor, which has been resolved in that structure, locates sidewise, intruding the lipid bilayer and positioning the glycine 2 here, the very end terminus, it's the external entrance of that tunnel. And then there's the black domain, and that wedges inside this translocation tunnel where the bile acids are transported through, deeply intruding into the protein and making several specific contacts that irreversibly engage this molecule. And then interestingly, there is that string domain, and we know that's very important for the activity of the bulevirtide, that covers the surface of that block and bridges the extracellular surface of NTCP, clasping that molecule and keeping it in one conformation. And this graph should just show you that binding of the peptide to the receptor, and this is an overlay of the native structure versus the bound structure of NTCP, displaces two core alpha helices or more core alpha helices, and that results in a shift of the panel domain and blocking the bile acid and sodium binding. So the point is that there is no sodium binding anymore. The receptor is irreversibly defunctionalized through that activity. Now, this was all theory. I cannot go into clinical data. The big question arises, of course, what does a bulevirtide entry inhibitor treatment is doing? And I just summarize very shortly the clinical findings here. If you treat patients, and these are 15 patients, this was the MER203 studies, these are delta patients, this is delta RNA, with a monotherapy of Hepludex alone, or MURB alone, you see a continuous decline in viremia. If you treat with interferon, you also see a decline of viremia, but it's not so profound. It's beneficial, but not so profound. The interesting observation here was that the combination of treatment of PEG interferon with bulevirtide has a very profound synergistic effect. So what we are asking for is, to what extent can an entry inhibitor as a combination therapy lead to maybe a functional cure for delta, and to what extent does a monotherapy help to maintain and control the virus? And this is the results of a recent study that should make us very hopeful for the future. What we had in the 204 study, just to remind you, that has been presented just recently at the EASL meeting. If you continue a co-treatment of PEG interferon with bulevirtide treatment for another 2 years alone, we get about 46% of patients that remained HDV RNA negative for 48 weeks after end of treatment in this 10 milligram bulevirtide group. This is this bar here, whereas the monotherapy is less efficient with curative therapy, and that's end of treatment 48 weeks. So there is a potential for curative treatment of eliminating HDV. This will be discussed in the future, to what extent monotherapy can help and combination therapy. That's not my task to do that, but what I want to end up in the second part is to show you novel approaches to address HBV or HDV entry, because entry inhibition seems to be a very important part of any treatment regimen. And I would like to start to say the problems with bulevirtide. It's effective. It's standard of care in Europe and Russia to treat chronic HDV infection, but it's complex high molecular weight lipopeptide. It requires daily subcutaneous application. It blocks entry efficiently, but also NTCP-mediated bile salt transport. This seems to be asymptomatic, or at least only elevated, but it would be nice to have some different things that do not interfere with bile acid transport. So the aim would be to develop smaller molecules with comparable specific activity and to develop orally available drugs with high potency and comparable pharmacokinetic behavior. I'll show you one unpublished approach that we followed. So what we did, we used the plug domain I just mentioned that contains the NTCP essential binding site. But this is much less potent and faster uptaken into hepatocytes. But what we did, we fused them to bile acids. We know that the bile acids interfere with HDV infection with very low efficacy, micromolar range. But what if we fuse the bile acid to that plug with a specific linker? And that's what's shown here. This is the peptidic fraction alone of the small plug. The bile acids would be somewhere here, but we can improve the efficacy of these conjugates to the level of hepgludex or the bulugurtide. They are much smaller, they are chemicals, and they can be maybe, and that's what we investigate, given orally. This is the pharmacokinetics, which is similar to the bulugurtide in mice, at least. There's no clinical trial yet here. We're trying to go for that. But there are other approaches. This is an approach I would like to share with you from Assembly Bio. And what they've done, they screened a library with low molecular weight entry inhibitors in Hep2 NTCP cells for anti-HBV and Delta virus activity. And they had a small molecule here with an IC50 of 29 nanomolar. This is active against all HBV genotypes, similar, which you would expect because it targets as well NTCP, and it's selective towards NTCP. So this is not so efficient against other bile acid transporter. It can interfere with pre-S bindings. So obviously, it targets the same tunnel that I just showed you, and it inhibits also NTCP bile acid transport. This is now in phase 1, is planned. Preclinical studies, I think, performed already, and we will see what comes out of that approach from Assembly Bio. Another approach was an academic approach from Stan van der Graaf. He's an expert in NTCP for a long time, and he cooperated with Johan Nates and also Philipp Möllemann. And they identified an NTCP bile salt uptake inhibitor. They screened for bile salt uptake inhibitors in order to treat cholestatic diseases. And they also looked for the HBV-HDV entry inhibition. And they found a substance shown here that is potent, 28 nanomolar, 60 nanomolar, for HBV or HDV infection. And they could show the antiviral efficacy here in Hep2 NTCP cells for B virus and for the Delta virus. And together with Philipp Möllemann, they looked whether that molecule can be orally delivered to mice, to PHH transplanted mice, and that worked. So this is an oral dose given at day -1. And then HBV infection was initiated, and by 100 milligram per kilogram, they could prevent the infection. So this is the first oral entry inhibitor that is able to prevent infection, whether it is as good as hepgludex and can also prevent the spreading in the liver. We will see, but this is another nice example. Let me go slowly to the end. I would try to show you 2 examples of antibodies. I already mentioned the HBICs that have been used for interrupting the perinatal transmission for a long time. They are HBICs, and they are novel antibodies that are now, and you heard it already from Sean at the beginning, from Virology, but there are other companies as well that develop S-specific antibodies that interfere with the heparan sulfate proteoglycans, but there are also pre-S antibodies that are targeting the pre-S domain that are in the region of the hepgludex. And this is some summary of the Vir approach, and the antibody is already called tobevibart. So there is a new name, and there have been recently clinical data presented. This targets a conserved conformational epitope of the S antigenic loop. It blocks intrahepatic HBV and HDV spread in humanized liver mice very efficiently, and the SOLIST phase 2 trial of that antibody showed efficacy on HBs complexation, also HDV serum RNA reduction, in patients, and it has already been mentioned that the interesting finding is here that we have a strong synergism with siRNA approaches that lowers the S antigen in hepatitis delta infected patient. I mentioned that there is another antibody, and that there are antibodies against the pre-S. I don't have the time to show you, and I want to sum up with my messages. So entry inhibition is an efficient way to block de novo HPV cccDNA formation, but also HPV cRNA, as I sometimes call it, replication, and also HPV integration. So double-strand linear DNA is also blocked. Virological responses, that's important to understand also for the discussion of primary endpoint, reflect the continuous decline of infected hepatocytes. So it's an indirect antiviral effect. It's not a direct interference with the replication cycle of any of the two viruses. Bulevirtide shows efficacy as monotherapy and has curative potential in combination with interferon in HPV co-infected patients. At least sustained virological responses over 2 years. We will see what happens later on, if there's rebound still possible, if S antigen is not eliminated. NTCP addressing drugs should be selective. They should be irreversibly inactivating the receptor, and they should be not transported. Remember that that bile acids are transported within milliseconds through the receptor, and that we have hours where we have an inactivated receptor by these peptidic drugs. Beside bulevirtide, small NTCP targeting molecules are under preclinical development. Conjugates, as I show you, orally available molecules. Antibodies that address the S domain, like the Vil antibody or the pre-S1 domain, show very good antiviral activity in chronic HDV patients. We also see the B trials here, and we will see in which way of combinations they will work. We know that synergistic effects of entry inhibitor are expected, generally seen, with drugs that prevent, for example, the cell division-mediated spread of hepatitis delta virus. This is something you don't address with an entry inhibitor, or with drugs that induce killing of infected cells, so some kind of immune modulatory events. That is, I think, what I would predict to be synergistic to bulevirtide. And the synergistic effect that we see with interferon in chronic hepatitis delta virus should maybe also be elucidated in chronic HPV patients. This is something that we are still lacking too many data on HPV trials. And with that, I would like to thank you for your attention and would like to acknowledge my group, which you can see here, and also people from Walter Meer's group, who did a lot of work on the pharmacokinetics, and of course, Ralf Bartenschlager, who's the head of the department, and for continuous discussion. Thank you very much for your attention.
Thank you very much, Stephan.